Welcome one and all. Today we are going to know about cytokines. Now what is this cytokines? Cell movement is cytokines. Cyto means a cell and kines means a movement. It is a broad and loosely category of small proteins that are important in cell signaling. They are released by the cell and affect the behavior of the other cell and sometimes the releasing cell itself. They include chemokines, interferon, interleukin, lymphokine, tumor necrosis factor, but they generally not hormones or growth factor are included in this. They are produced by a broad range of cell, including immune cell like macrophages, beta lymphocyte, T lymphocyte, and mast cells, as well as endothelial cells, fibroblast, and various other kinds of cell, for example stromal cell. A given cytokine may produce by more than one type of cell. They act through the receptor and are especially important in the immune system. They modulate the balance between the humoral and cell-based immune response. They regulate the maturation, growth, and responsiveness of a particular cell population. Some cytokine enhance or inhibit the action of other cytokine in the complex way. They are different from hormones, which are also important in cell signaling molecule in that hormone. The hormones circulate its much lower concentration and hormone tend to be more specific kind of cell. They are made up of specific kind of cell. They are important in health and diseases, specifically in the host response to infection, immune response, inflammation, trauma, cancer and reproduction. Now let's see the discovery of cytokine. Interferon alpha and interferon type 1 was identified in 1957 as a protein that interfered with viral replication. And the activity of this interferon gamma, the sole member of the interferon type 2 class, was described in 1965, and this was the first identified lymphocytic derivative mediator. Macrophage migration inhibitory factor, that is the MIF, it was identified simultaneously in 1966 by John David and Barry Bloom. In 1969, another scientist known as the Duedel Dimondi proposed that the term lymphokines, and to describe this protein secretion from lymphocytes and later protein derivative from the macrophages and monocytes in a cultural, and it was called the monokines. As scientists learn more, it was understood that these protein and other are part of the broad class of protein involved in the self-defense and should be called as the cytokines. Different from hormones, these classic hormones circulate in nanomoles, that is 10 raised to minus 9 molar concentration that usually vary by less than one order of magnitude. If we see in contrast, some cytokines such as IL-6 circulate in picomolar, that is the 10 raised to minus 12 molar concentration that can increase up to 1000 fold, especially during the trauma or infection. The widespread distribution of cellular sources for cytokine may be further than differentiated them from the hormones. Virtually all nucleated cell, but especially endo and epithelial cell, are residental macrophages and may near the interface with the external environment and are potent producer of IL-1 and IL-6 and TNF alpha. If we see in contrast the classical hormones such as insulin, they are secreted from discrete glands, example the pancreas. As of 2008, the current terminology refers to cytokine as immunomodulating agents. Part of the difficulty with distinguishing the cytokines from the hormone is that some of the immunomodulation effect of cytokine are systematic rather than local. For instance, To use hormonal terminology, the action of cytokine may be autocrine or paracrine in chemotaxis or chemokines, and endocrine as a pyrogen. Further, a molecular cytokine are not limited to their immunomodulatory roles. Nomenclature: cytokines have been classed as lymphokines, interleukins, and chemokines based on their presumed functions, cell of secretion or target of action. Because cytokines are characterized by considerable, the term interleukin was initially used by researchers for those cytokines whose presumed target are principally leukocytes. It is now used largely for designing of a new cytokine molecule discovered every day and bear little relation to the presumed function. The vast majority of these are produced by the helper T cells or T helper cells. Lymphokines produce lymphocyte. Monokines produce the monocytes. Interferon involved in the antiviral responses. And colony stimulation factors support the growth of the cell and semi-solid media. Chemokine mediated chemoattraction, that is the chemotaxis between the cell. Now let's know about the classification of the cytokine. Structural and functional classification are the two types. First, we will know the structural classification. Structural homogeneity has been able to partially distinguish between the cytokine that does not demonstrate a considerable degree of redundancy, so that they can be classified into four types. The 4 alpha helix bundle family member cytokine have three-dimensional structure with 4 bundle of several non-immunological cytokine, including EPO, that is the erythropoietin, and the thrombopoietin, that is TPO. Furthermore, 4 alpha helix bundle cytokine can be grouped into a long chain and a short chain cytokine. The IL-1 family, which is the primarily including the IL-1 and IL-18, and the IL-17 family, which has yet to be completely characterized through membrane cytokine, have a specific effect in promoting proliferation of the T cell that cause the cytokine effect. 15 knock cytokine include a member of the transforming growth factor beta superfamily, including TGF beta 1, TGF beta 2 and 3. Functional classification. Let's know about the functional classification of the cytokine. A classification that prove more useful in clinical and experimental practices outside of structural biology divides immunological response type 1 INF gamma, TNF alpha, etc. and type 2 is the TGF beta and IL4, the IL10 and IL13, etc., which also favors the antibody responses. A key focus on interest has been the cytokine in one of these two subsets tend to inhibit the effect of other in the other. The dysregulation of this tendency is under intensive study for its possible role in the pathogenesis of autoimmune diseases. JAK-STAT pathway. JAK-STAT is Janus kinase signaling transducer and activator of transcription pathway. It is crucial signaling mechanism that allows cell to respond quickly to cytokine and growth factor. It plays a major role in the immune response, inflammation, cell growth and differentiation. So cytokine bind to its receptor. A cytokine or the growth factor, example interferon IL-6 or EPO, bind to its specific receptor on the cell membrane. This receptor is linked to the JAK enzyme, that is the Janus kinesis, and inside the cell, and this JAK activation takes place. Phosphorylation begins. JAK protein are inactivated until a cytokine bind to the receptor. Once activated, JAK phosphorylates, add the phosphate group to the receptor, creating a docking site for the stat proteins. That is the stat activation takes place. Signal relaying begins. The stat signaling transducer or an activator of the transcription protein binds to the phosphorylated receptor. The JAK enzyme then phosphorylate stat protein and activating them. Then stat dimerization takes place and nuclear transport takes place. So the phosphorylated stat pairs up, dimerize and move into the nucleus. Then gene activations takes place, cell response begins. Inside the nucleus, the stat binds to the specific DNA sequence, turning the gene on or off, and this leads to the production of the protein that regulate the immune response, cell growth and inflammation. What is the function of this JAK-STAT pathway? Immune response. Help the immune cells fighting against the infection. Interferon signaling. Inflammation regulates cytokine responses, that is interleukin 6 and interleukin 12 signaling, and in blood cell production also it stimulates the red blood cells, that is RBC formation, and in cell growth and repair it controls the tissue regeneration, the growth hormone and GH. Disease linked to the JAK-STAT dysregulation. Autoimmune disease overactive leads to the arthritis and other diseases, cancer, and mutation in JAK-STAT cause uncontrollable cell growth, leukemia, and lymphoma. Immunodeficiency defects in the JAK-STAT weaken the immune responses. JAK inhibitors. Certain drugs target this pathway to treat diseases. Here are the names of the drugs. Tofactinib, used for the arthritis, and roxolitinib, used for blood cancer. Myelofibrosis. If we summarize the JAK-STAT pathway, the JAK-STAT pathway is a direct, fast signaling route from the cell membrane to the nucleus. It plays a key role in immunity, inflammation, and cell growth. Dysregulation can lead to the autoimmune diseases, cancer, and immune deficiency. JAK inhibitor help in treating condition caused by an overactive pathway. JAK-STAT pathway cytokine feedback inhibition primarily occurs through the upregulation of suppression of cytokine signaling SOCS protein, which act as a negative regulator by directly inhibiting the JAK kinase and preventing further STAT activity. So effectively limiting the cytokine signaling cascade. Once a certain level of cytokine stimulation is reached, this mechanism helps in maintaining a balanced immune response. The SOCS protein as a key regulator. When a cytokine binds to its receptor and activates the JAK-STAT pathway, the activated STAT, that is STAT protein, translocate to the nucleus and induce the transcription of SOX gene. The mechanism of SOX inhibition. The SOC protein contain a conserved domain called the SOX box, which allow them to interact with the E3 ubiquitin ligase complex, leading to ubiquitination and degradation of JAK or the cytokine receptor, effectively down-regulating the signaling pathway. Then different SOC protein can target specific cytokine receptor and JAKs, allowing for the fine-tuned regulation of cytokine signaling, and this negative feedback loop is crucial for preventing excessive immune response and maintaining homeostasis within the body. Other mechanisms contribute the cytokine feedback inhibition in the JAK-STAT pathway. Protein tyrosine phosphatase, that is the PTPs. Certain PTPs can dephosphorylate JAKs and STAT, further reducing their activity. And then protein inhibitor of activated STAT, that is the PIAS protein, can bind to the phosphorylated STAT and prevent their nuclear translocation, inhibiting the gene transcription. Cytokines are small signaling proteins secreted by immune cell and other cell to regulate immune responses, inflammation, cell communication, and tissue homeostasis. They act by binding to the cytokine receptor on target cell, triggering intercellular signaling pathway. There are various types of cytokine. Interleukins regulate immune cell differentiation, activation, and communication. Interleukin 1 promotes the inflammation and fever. IL-2, that is the interleukin 2, stimulate T cell proliferation. Interleukin 6 drive acute phase response and inflammation. Interleukin 10 anti-inflammatory cytokines suppress immune response. Then interferons, they protect against viral infection and modulate immune responses, that they are known as IFNs. And example, interferon N alpha and interferon N beta antiviral response activate immune cells, and interferon N gamma activates macrophage enhance antigen presentations. Tumor necrosis factor, they regulate inflammations, apoptosis and immune cell activity. The TNF, that are the tumor necrosis factor alpha, they promote inflammation involved in autoimmune diseases. Colony stimulating factor, they are known as CSF, and they stimulate the production of blood cell in the bone marrow. Example, GCSF promoter neutrophil production, and MCSF stimulate macrophage production. Chemokines, they direct the cell migration, chemotaxis, especially the immune cell to infect the site. Example, the CXCL8, interleukin 8 attract neutrophil to infect the site, and CCL2, MCP1, that they are the recruiters of monocytes and macrophages. The transforming growth factors, they regulate the immune suppression, tissue repair and fibrosis. Example, TGF-beta inhibits the immune response and promote tissue healing. The functions of cytokines are immune activation, stimulate T cells, beta cells and macrophage, inflammation regulation, interleukin 1, TNF alpha and interleukin 6 trigger inflammation, antiviral defense. The IFNs inhibit viral replication. Hematopoiesis, CSF, stimulate blood cell production and wound healing and fibrosis. In that, the TGF-beta promote the tissue repair. The cytokine related diseases are excess of cytokine activity causes the autoimmune diseases. Example, arthritis and lumps. And if we see the cytokine is strong, then overproduction of the cytokine in several infection, example COVID-19 and sepsis. If we see cytokine deficiency, then immune suppression leads to infection or cancer progression. Thanks for watching.